Okay, I guess with 5 minutes of delay we can start.
No problem.
Welcome everyone to the first of these 10, I suppose, seminar series. There will be one each month. Many of you are in the mailing list that advertise on the seminar. If not, you can subscribe just writing me. Some of you have been last year at seminar at Interrogate. This year the format is slightly different. We have both presence and online, and that's the reason of our technical problems. But I'm not going to bother you further, and I start to introduce briefly our speaker, Professor Guido Sanguinetti, with just a few words. Guido studied physics at the University of Genova, then moved for a PhD field in Oxford in mathematics, and later on he held the positions both at Sheffield University and later in Edinburgh, and in the field of bioinformatics, as many of you know, and of course machine learning. And 2 years ago, around 2 years ago, he moved back to Trieste, where now he's a full professor in applied physics at SISSA and also chair of data science. And more recently, he's going to become scientific director of the newly created Data Science Hub in Trieste.
Yeah.
And I leave the floor to Guido.
Thank you very much, Alberto, for a very kind introduction. And let me start going into full screen. That's a particular, and I'll take the mask off now so that my nose and ears suffer less. And it's a real pleasure to be here. As I was telling the guys, the last seminar in presence was at ICGB, and I remember everyone was very surprised that the Chinese were taking draconian measures in Wuhan for what seemed to be, just a slightly worse flu. It turned out not to be the case, of course, and it was a lot worse than a flu. But in any way, it feels as if I'm picking up exactly where I left it, giving a talk at area, as I was doing nearly 2 years ago. And in fact, actually, and I do apologize, some of the content of this talk was also in the last talk at area. So some of you may have seen some of these, but we have progressed a little bit since, and I'm very pleased to show you some more recent results that improve and generalize on what we were doing at the other time. So I've been instructed to have that every seminar must have at least 20-25 minutes that are sufficiently general, and I don't know whether mine would be 20-25 minutes or whether they would be sufficiently general, but I hope so. But essentially, so can you see my pointer on the? Yes, okay, I can use that for indicating on the screen. So I'm going to start with some considerations that are nearly philosophical, so they're totally general. And then I'll give you some introduction on the actual the data generation procedures, which is kind of a key to understand what methods are suitable. And I'll give you also some introduction to Bayesian statistical methods, which is the bread and butter of what I do, particularly with respect to this talk. Then point 4, the first half of point 4 is what people might have seen already, BRI, this Bayesian regression for, oh no, what's it done? It's gone forward now, I don't want to do that. Well, okay. So it's very sensitive, your touchpad, I noticed. So BRI is what you will have heard before, but we have now a newer version, which is in some way fairly different and does many more things, which came out just, I think, last month or a couple of months ago. And then I'll give you also a flavor of other applications of Bayesian methods in single-cell omics, particularly in the field of DNA methylation. And again, this SCMet paper is a very recent paper that came out at the beginning of the year in genome biology. So let's start with the more philosophical part. I mean, I imagine actually some of you take big data to be something that was not new, but has always been with you essentially. But about 10 years ago, the name big data started to circulate in academic environments. And so this was a conversation that I had with a fairly well-known statistician in the UK about 10 years ago, when everyone was very upset about this big data frenzy that was populating funders and so on. Everyone seemed to be crazy about big data. And the question my colleague pointed out was, what is the point of having 1 billion IID data points? So IID means independent and identically distributed data. And the point he had was perfectly fine, yeah, because if you're interested in collecting some basic statistics, I don't know, if you want to estimate the average height of the population in Friuli, you probably get a pretty accurate estimate if you subsample randomly 150 or 200 individuals, or if you systematically collect 1.2 million individuals. So his answer was, if all we want to know is do very simple statistics, then big data is, the solution to big data is throw away most of it and just do your easy calculations on a subsample. And this is not so stupid as it may seem. So I remember roughly at that time, one of the questions that were fairly big in machine learning was how to make Gaussian processes more scalable. Gaussian processes are a kernel machine for regression and classification. And I remember a beautiful talk by my former colleague in Edinburgh, Chris Williams, who was comparing several methods for scaling better Gaussian processes. Clever methods, matrix reduce rank approximations, some of them fairly sophisticated. And then the baseline was randomly throw away data. And it wasn't so much worse than doing anything clever. Yeah, so unless your question is really needs the big data, and there are some questions that need big data, now we understand them better. I think at least 10 years ago, it was a good question. Why do we need big data? Why do we have to collect data about everything? Now, the caveat, and I think the answer that I could now give to that friend, but I couldn't at the time because I hadn't had enough experience, was, these kind of questions, you're right, big data is no big deal if your data is low dimensional. If your data is very high dimensional, and you want to start to estimate covariances, for example, then immediately things become more problematic. Why? Well, suppose to have even Gaussian distributed data, but now your data is not one-dimensional, so you don't need to estimate a mean and a variance, but is 1,000-dimensional, so you need to estimate 1,000 mean parameters, and a full covariance matrix, which contains roughly 500,000 parameters. Well, you're not going to be able to do that with a subsample of 100 data points. That's pretty clear. Even so, though, even if the data is high dimensional, the big data, some sense conceptual failure that was not resolved until recently, at least intuitively for me, was, will you ever have, if you're really in high dimensions, will you ever have sufficiently high dimensional data? If we think about images, images, hundreds of thousands of dimensions. If you are trying to approximate a function on hundreds of thousands of dimensions to a certain, precision, for example, that's the problem, that's the regression problem. And learning a function from an input space to a scalar, if your input space is 100,000 dimensional and you want to approximate the function through evaluations at finite set of points, so that the true function in your approximation is within epsilon, the number of points that you need is of the order exponential in 100,000. So there's no way you will ever get enough data to do that. We can't even imagine, even, I don't know, with quantum computers, we probably can't store that amount of data. What we have learned, I think, since in the last 10 years is that there are quite a few domains where data may be high dimensional, or at least may be presented to you as high dimensional, but there are very informative low dimensional representations. And that is really the case where big data, big IID data becomes really useful. So I'm thinking here of the whole representation learning field, so all neural networks, where you can basically extract features that live in a much smaller dimensional subspace or manifold of your input space and are still highly predictive. So they capture somewhat the essential directions of variation. And this is really the field. So I think, 10 years ago, where neural networks were just a curiosity, essentially, my statistician friend probably had a point, what is all this about big data? But now we kind of understand that if you want to approximate complicated functions, you need data that grows and you need models that grow in a controlled way. So that's why I say it's increasingly clear also mathematically why really big data enables you to go further in these cases. Having said big, there is still one thing that I'm very much against in the statement of my friend. What is the point of having 1 billion IID data points? Now, the point is the IID is a very difficult assumption to verify in practice. Now, if you were to kind of consider a joint distribution over all possible features, measurable and known, then, okay, everything would be obviously IID. And I think that is the underlying assumption in neural networks, methods, and so on. But in many cases, what matters are exactly the differences that exist between different data points. So in many cases, IID is really a bad assumption. And then in those cases where IID is contrary to your goals as a scientist, you probably should think differently in terms of the computational strategies. So let's take, for example, some examples where obviously IID doesn't matter. Okay, does matter, sorry, not doesn't matter. Now, you guys are working a lot with transformers, and one of the beautiful things that transformers do is generate text. That makes a lot of sense, well, or seems to make sense. How do they do that? They get a lot of data from text that exists around, and they learn structures about sequences of words, and they learn it in a very clever way. But now, if you wanted to, say, generate text that is in the style of Victor Hugo, for example, maybe with Victor Hugo was famously prolix, you might get a sufficiently large data set, but otherwise, I'm pretty sure that a transformer trained on a big bank that contains many, many different authors would never be able to replicate a style, for example. Maybe a more relevant question, and this is actually a commercially relevant question, is machines that are used routinely in hospitals to take scans of patients. Now you want to use image recognition or image classification systems to deal with these images. Unfortunately, hospitals have different machines, so different brands, different models, and also typically they will have slightly different settings and so on. So even though we have potentially an enormously large set of, I don't know, breast cancer scans, and I say breast cancer because I know someone who worked on breast cancer scans, then actually the set that comes from one hospital with that setting is a fairly limited set. And that is a problem. That is a problem because you can't, unless you do something clever, you can't assume that predictions that are trained on a broad set of different machines and so on will actually be effective also on that specific machine for that patient, which is what you care about. Similar question, you want to do prognosis by taking physiological or blood samples and so on. Well, every person is slightly different, so they're not all IID. Humans, they're not IID. There are many characteristics, some of which you can observe, for example, age, gender, ethnic background, and so on, and they all could explain part of the variation. So the key thing is, if you manage to explain more variation, you'll be able to do from external variables that you can measure, then you'll be able to do better with less data on the tasks that are difficult. The question that will be more relevant for today's talk, because it's a talk in genomics, is gene expression or other molecular values in individual cells. So we have these technologies. We are made up of a few trillion cells. All of our cells are very similar in some ways. They all have roughly exactly the same DNA, except for our skin cells that are subject to continuous bombardment from UVB. But they're not all identical. So what makes my liver cell different from my neuron, from my muscle cell, and so on? Well, we don't really exactly know. I think biologists don't exactly know. But what they can do, they can now measure a lot of properties of cells, either in large collections of cells. So, take a whole liver from a mouse, mash it all up, and take all the gene expression from that liver in that mouse. But increasingly, also they can measure from each individual cell. And you start learning fascinating things like, the liver is not made up of all homogeneous cells. There are cell subpopulations. You can try to understand what the molecular determines in terms of the function. So cells that produce certain proteins do different things. And how much variability is there in these productions and so on. This is the topic of today's talk. So the technologies that produce this data are relatively young, and they all have to do with measuring molecular phenotypes, if you wish, of individual cells. So we know that DNA contains the genetic information of each cell. So somewhat it's called, it's like, the hard disk of the cell is where the programs are. But what actually does things in cells, so the machines that catalyze the reactions, that give the cell its structure, that keep the DNA wrapped around the histones and the histones themselves and so on, they're all proteins. Yeah? So proteins are the machines. DNA is so proteins are like the robots, if you wish. DNA is the hard disk. Each robot does a slightly different thing. So in some sense, some parts of the DNA are executed, but not all, in the same way in different cells. So same DNA can produce different proteins. And the process of protein production is tightly regulated. It's called gene expression, the process that leads from the genetic material to the protein, is tightly regulated in order to preserve the cell identity and the cell function. It's a two-step process. First consists of an intermediate, RNA, and then that's a process called transcription, and then RNA is translated into protein. Naturally, two questions. This is a static picture, but we have a lot of different types of cells, as I pointed out. There are also situations in which cells have to react in time. So there must be dynamics. And how do these dynamics work, essentially? And the second question, which is closely related, of course, is what can we measure within cells? It turns out we can measure a lot about DNA and RNA, much less about proteins, although things are improving. So most technologies are about measuring the total RNA inside a cell, or sequencing the DNA inside a cell, or sequencing the DNA in particular chemical modification states. The measuring of the RNA is called transcriptomic, and it's a fairly advanced techniques which relies on a number of different advanced technological features. Well, first of all, you have to isolate bits of tissue, then you have to separate the cells. This is something that cells really do not like. So if you are a liver cell, you are very well used to be next to your friend liver cells, and you really don't like being separated from that. So within this process, you are introducing massive biases, because cells, you will see signals of cell stress. Some cells die, so they burst and you don't measure anything anymore, and so on and so forth. But the dissociation and single cell isolation is a problematic thing, but it is done. Then the single cell goes through one cell at a time through a microfluidic device, and eventually gets isolated, either put in a well on its own or put in a droplet. And you break it, you take the genetic material, so the RNA or the DNA, in case of the transcriptomic is the RNA, you reverse transcribe the RNA to get little bits of DNA, and you sequence them. In this process, you're going to lose, it's a single cell, so there is already not an enormous amount of material. You're likely going to lose most of it. Yeah? So it's a subset of the RNA in the cell that you actually measure. People do also different things, as I was saying. We'll see later about DNA methylation, which is another thing that you can do to single cells. By the way, I should say, the people that are listening from Zoom, if you have a question, just unmute and ask, because it's quite easy for me to miss questions in the chat. If you have questions that you want to ask at the end, put them in the chat and so on. But if you want to have some clarifications, so to speak, during the talk, just stop me and ask. So the kind of questions we want to ask from the single cell type of data, we have large amounts these days of cells. So we can take out and sequence the content of a few thousand or a few tens of thousands of cells individually. If you think of it as a matrix, we have 20,000 genes, maybe similar number of cells. We observe a large amount of variability. So we see that the same gene in some cells is absent, in some cells there's quite a few of it, some cells there is a little bit, and so on and so forth. But some of this variability is purely noise. Simply, for example, for the fact that we probably lose at least 80% of the content of the cell RNA during the process of breaking. Some of it is artifact, because, as we said, the cells are very strongly stressed. Some of it is variance that we may be interested in. Maybe the cells, the individual cells that we're measuring, are from two different conditions, from, say, patient with a disease and a normal patient, or they are from the cancer and the surrounding tissue. So that's variance that we're interested in, because that variance might be linked to the biological or medical process that we're trying to investigate. Some other variance might actually be variance that is truly biological. For example, different cells might be in different stages of the cell cycle, but it's confounding. We don't care about that, because what we care is what are the hallmarks, say, of the disease, not whether it's in stage G1 or M or whatever. So the whole point and the question that we're trying to answer with these types of methods is, we obtain data, which is highly noisy. We see variance in this data, and we would like to be able to say, this variance is the technical noise. This variance is artifact of the experiment because we stress the cells. This variance is confounding variance because the cells are in different cell state. This variance is what we care about. This gene changes when the cell has this disease. So decomposing variance is basically what we do. And in this case, it's important to exactly understand what are the shared components of the variance. All cells that have the disease have these particular features. And what are the individual components? So it's a clear case where we can't just say everything is the same distribution. We have to model exactly what are the differences between the distributions conditioned on the disease case and conditioned on the healthy case. And that's what Bayesian methods do. Okay, so I wonder whether I can move these slightly further up. Yeah. Yes, so these I can make smaller, and then probably I can move. That's probably optimal now. Good. So Bayesian methods are a way to model joint probability distributions. In fact, I mean, what I do here is not necessarily Bayesian. I just model probabilistically a system as a collection of random variables, and then I say, okay, now that I observe some random variables, conditioning on these random variables, what does this tell me on the other distributions? So these are incredibly basics. They are the fundamental rules of probability. We have a joint probability. We're interested in systems that presumably have some degree of complexity. I think complex systems must be constituted of at least two parts, otherwise it's difficult to think of complexity. And so for the purposes of the introduction, we think of two random variables, but a random variable could be y, for example, could be a collection of random variables and have a non-trivial structure. So we talk about their joint distribution and we talk about their conditional distribution. What does it mean? I have two random variables. They can take they're variables, so they can take certain values. Joint distribution is the probability of x being I and y being j. Conditional is if I have observed or I know that y is j, what is the probability of x? It's deeply connected with the concept of information. So how much information does the y variable tell me about the x variable? Okay. The fundamental idea and the reason why Thomas Bayes is famous is that this is a way to not only connect information flows, but invert information flows. Now, if I have a certain prior belief about what y should be, and maybe this belief is a complex model that says about how the various bits of y, how they interact, and I observe something about y, so I have a model of x, which is what I can observe, depending, conditioned on y, then Bayes tells me, okay, now I can invert the information flow, not only from y to x, but compute what can I tell now about y, given that I've observed x. So this part is my model of what y should be like, because I have a conceptual picture of how the various bits of y interact, and what x should be like given y, because, for example, this is the model of my instrument. This is the reverse information flow. I start from what I've observed. What implications does it have for what I could not observe? Very, very simple. But the problem for any model of any decent complexity, actually computing these things is very hard. And what is difficult to compute is this normalization constant in general. The reality is, in this talk, I'm not going to, there is this, probably the vast amount of research is about efficient ways to compute these kind of normalizations and invert these probability laws. That's most of Bayesian research. I'm not going to mention that. What I'm going to tell you instead is how we use these concepts and how we build models of y and x when y is a biological process of interest, like splicing in gene expression, and x is the thing that we can actually observe, so short segments of reverse transcribed RNA. A nice way to explain models is the so-called graphical model. So I'll show you an example. Each random variable is denoted as a circle. And if it is observed, I color it in. An arrow denotes a conditional distribution. Okay? So the distribution of my observations R depends explicitly on this unobserved random variable I. The plates represents group of variables that are identically distributed. So for example, here, what I'm saying is that I have a group of n subset k random variables I, n, and each of them has got its associated observation Rn. Also, I have k groups, and each group is homogeneous. And what this tells me is that each group has got a set of observations x that influence a latent variable psi, k. And then at the top of the hierarchy, if you wish, there are some variables that are global. So they're common to all groups and all realizations. So that's the way to think of it, you know. I can generate a sigma, a lambda, and a w. Then I have a group of random variables which have an observations attached, which are the xk observations, and merging these xk with this global variable, I produce a local variable pck. Then given a pck, I generate n replicate random variables in, which each of them has its observation Rn. This is an example of a generative model, because if I know the distribution and I know what are the conditional distributions implicated by these arrows, I can simulate data, essentially. So this is an example of generative models. Some of you might be familiar with more complicated generative models, but the basic idea is exactly the same. The other thing that I want to tell you about, and that we will use heavily, was already implied in the previous graphical model, is the idea of hierarchical data specification. Yeah. So what do I mean? I mean that sometimes you can group variables hierarchically. For example, we could say we have a certain gene J, which is in a cell I, which comes from experiment K, which comes from condition L. So for example, I have two conditions, I don't know, healthy and cancer, and then I do 3 experiments in cancer and 3 experiments in healthy, and then each experiment has got 1,000 cells, and then within each cell, I have 20,000 genes. So this is an example of a hierarchical data specification, and the way that you will construct your models is basically saying, okay, the global variables are no longer relevant if I know, say, for example, in the gene level variable depend on the cell variable. But if I know the cell variable, I don't need to know the batch variable. So information somewhat has this Markovian property, yeah? Where if I condition on one level, then I can say everything about the lower levels, and I don't need to know anything about what is above. And the reason why they're particularly useful is, well, at least in my opinion, they're particularly useful, first of all, because they're 10 a penny, as the British would say. You know, this kind of specification of data as a hierarchy, this is entity I in group J, in supergroup L, and so on and so forth. This is everywhere. In every type of model of every system, you can come up with such a specification. But it's also useful because it gives you a natural way to reason about the various uncertainty. So you could say, if you have such a decomposition, what is exactly the variability between cells that are otherwise identical, they're in the same experiment? What is the variability that is presumably technical coming from different groups in the same condition? What is the interesting variability that comes from the different conditions and so on? So it's a very natural way to say how much of the noise comes from which level in the hierarchy, and therefore enables me exactly to answer the questions I had started with of breaking down variability. This is how I would represent the previous discussion in a graphical model. I observe the gene expression in cells and then batches, conditions, and so on. So that was the introductory part, which turned out to be almost half an hour, actually, so more than I was tasked to do. What I will do in the rest, I'll show you some applications. The majority of the work that goes into the applications is in 2 parts. One is devising the algorithms for doing inference, so for computing the Bayesian flow of information or reverse flow of information. And the second, and I have to be honest, the second big part of the work is actually preparing the data in a way that it becomes suitable, because these data, you know, there are large amounts of pre-processing that go into it. You have to throw away cells with fewer than a certain number of genes. You have to try to distinguish which cells were broke during the experiment and therefore they're completely unreliable and so on and so forth. I'm not going to talk at all about the hard work parts. I'm just going to tell you, this is the problem, this is how we model, these are the results that we get. So the easy part, the stuff that I do typically, the students do the hard part. So the first part is about splicing. Now, splicing, when I said that gene expression is a two steps, well, each of these steps is complicated. So the production of RNA itself is not just a simple process where polymerase slides around and produces RNA. There are many, many steps, and one of the most important ones, and this was a big discovery only 50 years ago, really, is that gene expression is not continuous. So you might have a gene which occupies a certain stretch of DNA, but the stuff in that, the subsequence that codes for the protein, is not the full stretch of DNA, is only a subset, and a discontinuous subset. So there is a bit that codes for the first part of the protein, then there is something in the middle, then there is another bit that codes for the second part of the protein, and so on and so forth. So there is this structure which is made up of exons and introns within most genes, at least in higher eukaryotes. So here's the typical picture. The polymerase, which is what does transcription, produces a pre-mRNA, it's called, an RNA that contains some bits that need to be thrown away. And it's made up of a sequence of exons, intron, exon, intron, exon. And the process of splicing is the process of cutting the, let's say, useless bits, although sometimes they do other things, so they're not useless, and putting together the protein. Now, if it were always the same, there would be little point for having evolved splicing. The reality of having splicing is that this gives us a combinatorial, well, gives the cell a combinatorial handle to produce a wider variety of proteins, okay? Because, for example, from this protein, which contains 3 exons, from this gene that contains 3 exons, using the same genetic stretch, you could, in principle, produce 2 to the 3 minus 1 different proteins. A short protein, only these, and then you could have a protein these and these, a protein this, and so on. 2 to the 3 minus 1, because the empty protein doesn't count. In reality, this combinatorial complexity is almost never realized, and the most common type of splicing variation is alternative or so-called exon skipping. So in one situation, the cell will produce proteins that are only the 2 dark exons put together, so shorter proteins, but in another situation, for example, in response to DNA damage, we produce another protein from the same gene, which contains all the 3 exons. And that partly explains why, even if we have only about 20,000 genes, there are about 200,000 proteins that are produced by human cells. Or at least that was the data until a while ago. I think we still discover new proteins on a weekly basis. It's a fascinating control of gene expression mechanism. It's not fully understood, but it's implicated in many diseases, from cancer to neurodegenerative diseases and so on. So one key question is, quantify if I have a gene that can produce 2 different proteins using this exon skipping mechanism. From measuring messenger RNA, can I say how much of the mRNA comes from one protein, will produce one protein, and how much will produce the other protein? And it's not so easy, because you guys that know a lot about genomics, most platforms, I mean, the vast majority of platforms, don't produce long sequences of DNA. They produce short reads. And therefore, you can see, this could be an example of an outcome of an RNA-seq experiment, two-ended, pair-ended experiment. Some of them, map entirely to exon 1. So you can't possibly know whether this read comes from the long or the short isoform, because both isoforms, so this is the name of the 2 transcripts that correspond to different proteins, they both contain exon 1. The informative reads are, for example, this one. Yeah? This one contains a junction between exon 1 and exon 2, and therefore this must come from the log isoform that contains also the middle exon. Or this one that contains a junction between exon 1 and exon 3, so we can be sure that the RNA, the transcript that contained that read, came from the short isoform. The problem is that, of course, the number of reads that overlap with the junction is a small fraction of the total number of reads. Yeah? And so the very earliest RNA-seq experiments on bulk, not on single cells, they had, I don't know, a few tens of millions reads. They would throw away, if they wanted to do splicing, you know, they could rely only on 1 million out of 30 million or something like that. And that was because the way to estimate splicing was essentially looking at counting junction reads. Now, around 10 years ago, in bulk, a major advance, which was made by all the main methods for RNA-seq, meso, cufflinks, and so on, and now the more common Callisto and so on, was to take a probabilistic point of view and say, okay, no, actually, the total population of reads is a mixture of reads coming from the short and the long isoform, and I model them probabilistically and get a posterior estimate of the ratio. And by doing that, they were able to show, this was the paper, the meso paper, Nature Methods 2010, that you would obtain much more confident estimates of the alternative splicing ratio. What does this tell me? So here they had a situation where they had exactly 50% of long and short isoform. And they could show that using the probabilistic method, they always got the right estimate and they got it with high confidence. While using the direct method, when you got to slightly lower coverage levels, you would get very high, possibly uselessly high confidence intervals. You weren't able to say, it could be anything between 30 and 70%, which is not a particularly useful piece of information. So probabilistic methods improved. They also have increased error bars. And presumably, if they did the experiment going further and zooming in on this bit between 0 and 250, also these error bars would become uselessly large. Now, where do we fall if we're thinking about single-cell RNA-seq? So these methods were developed before single-cell RNA-seq. They were developed for bulk RNA-seq. Single-cell RNA-seq, the average coverage for sparse-seq, which is, you know, the expensive technology, is somewhere here. Okay, so it's really around, if you're lucky, 50. So in this regime, presumably these error bars also for the probabilistic methods would be very large. That is actually the case. Yeah? So here is you see basically some characteristics of single-cell RNA-seq technologies, which make it particularly challenging. So the high noise at low levels of expression. So normalized read counts, as you see, the majority are around 10. So there we were looking at 250, the majority is around 10. And in fact, if you try to quantify splicing from single-cell gene expression data, you get disappointing results. Yeah? So this is another probabilistic method, which Yuan Hua, my student, developed beforehand for bulk. Here is, you know, you would expect that a single cell from a certain population would correlate well with a bulk from that population. And if you try to do splicing estimation with bulk methods, you get that almost all your estimates fall around 0.5, which means I don't know, essentially. And only a few give you relatively confident estimates of splicing. So here you would expect to see something that is along the diagonal. Instead, you see a big bar on 0.5. And if you compare two single cells, which are supposedly identical, of course, there will be some variability, but again, almost everything returns a 0.5 estimate. Nevertheless, there is some evidence that splicing also, alternative splicing also happens in individual cells. And here is a picture that looks at expression of one particular gene in individual cells. So each one of these is a single cell. These lines here are the junction reads. Yeah, so the reads that we can unambiguously assign to either the long or the short isoform. So for example, here we see that in this particular cell, there is evidence that there are both the long and the short isoform, because we have a certain number of reads that straddle the junctions. So it does exist biologically as a phenomenon. It's just that in many cases, we are in this situation where instead of having this cell was particularly lucky at about 20 junction reads, you have 1 or you have 0. So how are you going to solve this problem? Well, our idea was that, because we are Bayesian, there is the observation model, but there is also a prior model. And if your data is not strong enough, then maybe a prior model can tell you some useful information. Obviously, that is equivalent to knowing the answer before you pose the question, which is not a fair way of doing science. That's where learning comes in. So the idea is try to learn something informative, because on some genes, like in this one, you know something. You have good enough data for that gene, that cell. So try to learn the characteristics that predict inclusion of the exon, and then use that learning to impute. So the idea is when you have plenty of data, data speaks, and data teaches you also how you should expect things when you don't have that much data. What are the things that can predict splicing? Well, there were a few high-profile papers that show that genetic sequence actually is a good predictor of splicing. And so what we did, basically, we came up with a prior distribution on this probability of being included for an exon or not, which is regressed against genetic sequence. So in a certain cell, we're trying to learn what are the genetic signals that predict inclusion of the exon in that cell. And then we learn that from whatever data is available, and we use that information to generalize to all. That's the graphical model, which I have already described to you actually before. These are the details. Now, what you can do, you can start getting, Pearson correlations that are very much stronger between different cells that are supposedly identical. So we don't get that big blob in the middle. We get things that are roughly more correlated. And if you compare to many methods like Callisto and cufflinks, the correlations that we expect to be fairly high actually become reasonably high. We've also learned, so this essentially is a way to explain some of the variability using the genetic sequence. Another thing that one could think, so it's a way of transferring knowledge from highly expressed genes to low expressed genes by learning sequence features. One thing that one could also think is to transfer knowledge from one cell to another cell. Maybe some features of the cells are also predictive of splicing. What does that mean? Well, we basically want to know if there is some splicing events that are associated with some cell features, for example, developmental time or disease or something like that. So this gives us a way to share information between different cells and so impute, but it also gives us a way to quantify whether specific splicing events are associated with particular conditions of the cell. How do we do it? We increase the graphical model. So this is the part relative to Brie, as you've seen it before, but now we also have a feature. So before we had a feature associated with the gene, which was the sequence. Now we also have a feature associated with the cell, which is, for example, is this a disease cell, or is this a cell of a certain type, or is this a cell in early development or late development or something like that. So what we are saying is that essentially the probability of being included for an exon is something that depends on its sequence and also on the particular characteristics of the cell. And then we can look at these parameters alpha that tune how important is the cell type for the inclusion and say, okay, these ones have a particularly high alpha, so they represent genes that tend to have a significantly different splicing between different cell types. What can you do with BRI2, which is this extension? Well, for example, one of the examples in the paper, and there are more, is to try to detect differential splicing events which are associated with particular diseases. One data set we had was a mouse model of multiple sclerosis, and we were able to identify a fairly large number of genes that, or really genes of exons that are preferentially included in disease and preferentially excluded in normal cells, and so presumably associated with some of the mechanisms of disease. Now I have about 5 minutes left, and I'll tell you a little bit about conceptually similar ideas in a completely different application field. Well, completely different. It's still single cell, and it's still single cell omics, but it's not transcriptomics, it's DNA methylation. So, as I said, different cells have got the same DNA, but they don't have the same DNA in the same condition. So the DNA is never naked inside the cell. It always has, it's wrapped around histones, it's got conformational features, it's got chemical features as well. One of the chemical features is its methylation level. So the cytosine nucleotide, particularly when it is followed by a guanine, so in the CpG context, tends to be methylated. So this is the cytosine, and this is what happens when you attach a CH3 group to the cytosine. And this is still relatively poorly understood. So my friend Adrian Bird in Edinburgh wrote a paper in 1981 called The Function of DNA Methylation, which has many thousand citations. And I think he regrets the title of that paper because he's still trying to figure out what it is. But there are ways, I mean, it's understood to have something to do with gene expression in terms of controlling gene expression. And it can be measured. And now it can be measured in individual cells as well. So you basically will get reads, as with all systems, and in some reads you can tell, okay, this CpG that flows into this read was methylated or not. In the majority of cases, you can't say it because you don't have a read covering the CpG. So the data is high dimensional. I mean, in principle, we have 16 million CpGs in the human genome. So in principle, it's 16 million dimensional, and it's extremely sparse in that for up to 95% of these 16 million, we just don't have an observation. Now, the question that we would like to understand is whether, so this has prompted a large number of papers. Well, I'm citing here 2, which try to do imputation and so on and so forth. But what we want to answer here is instead trying to understand when we see variation between different cells in methylation, which variation is associated with different states of the cells. We're trying to decompose the variation in methylation across different levels. And that's how we do it. We again have a hierarchical model. So we have a bunch of top-level variables which control the mean and variation in methylation at a certain feature, at a certain CpG, intrinsic, let's say. And then we have explanations. So we have features that can explain part of the variation in terms of, so we have variables that explain part of the variation in terms of features of the cells and so on. In practice, what can we do? So we've taken a data set that was published a few years ago, which did measurements of DNA methylation in large numbers of brain cells. And the kind of things that you can do, you can basically see which regions of DNA are methylated in a different way between different types of cells, and presumably therefore are somewhat associated with the process of differentiation. So here we had excitatory and inhibitory neurons, and these were the label of the cell, and we were able to identify a large number of DNA regions that have different levels of methylation in cells that are inhibitory and in cells that are excitatory. And of course, then there are things that we can't quite well explain. So it looks like this particular gene is highly methylated in excitatory neurons, but there is a subpopulation of excitatory neurons that have intermediate levels of methylation. And what are they? Are they neurons that are transitioning between being an excitatory and inhibitory? I don't know. But this kind of analysis shows you that maybe that gene is one to look for if you're trying to understand what turns a neuron from inhibitory to excitatory. You can also use these methods for feature extraction, so find what are the most distinctive features and then use them for clustering, and we showed that it produces an improvement and so on, all the kind of things you would expect if a paper gets published. Of course, if we didn't get them, we would not have included them into the paper or would not have published the paper. So I think there's enormous amounts of excitement in the biological community about these single cell techniques, maybe comparable to the excitement that was in the statistical community about big data 10 years ago. And I think it's very high, it's very motivated, and it's well motivated. There are really interesting techniques that allow us to measure things that could not be measured. However, it has to be understood that the measurement process itself is complicated and comes on top of very complicated, complex biological processes. So there isn't a single source of variation that contributes to what we see in the observation. There are multiple sources of variation. And we need to be able to take them apart. Otherwise, I don't know, we're not going to be able to associate interesting features with interesting biological mechanisms. I think, and I'm not the only one to think that modeling using probability distributions along the lines of what I've shown you is a good principled way to do this kind of signal decomposition and to potentially be able to associate, for example, what I was showing you exactly was an example of that. Associate which variation is due to difference between excitatory and inhibitory neurons, as opposed to technical noise, or as opposed to the fact that the cells are stressed and so on and so forth. So this is a way to try to extract exactly that type of signal. It's also, and that's not something I've talked about, but maybe the next time I give a talk here after another pandemic, I'll have some solution to that question as well. We now have techniques that enable to measure simultaneously expression and DNA methylation, for example. So we could try to see not only what are the joint variations, but whether we can associate some mechanisms of variability in one case to variability in the other case. I'll stop here because I'm just out of time. I want to thank the people. So the work I presented is work primarily by two former PhD students, Yang Hua, who's now an assistant professor in Hong Kong, and Andreas, who's still in Edinburgh as a semi-independent fellowship, and it was in collaboration with my colleague Catalina in Edinburgh, and Riccardo Argelaguet, who was a former student in Cambridge and now is a postdoc at the MRC unit in biostatistics. And these are the references. The first is a semi-philosophical review paper, and then the others contain the actual technical work. So thank you very much, and happy to take any questions. From the floor or from the... So let's ask from here. And so can I see the...
Guido, I have a couple of questions. Probably ideal question because I'm not an expert in genomics and neither in the things that you are doing. But coming back to one of your slides, so that is actually the point where you say that there are just I don't remember one slide which you show the number of data that you are counting. Come back.
This.
Yeah, this one. This 19 is actually when you have the overlap between the two exons.
So exactly, in this particular cell, there were 19.
Exactly. And when you come back, go back once again. Back. Here, coverage, RPK. This means that in this isoform quantification, counting this isoform quantification, only 250 were overlapping. Is correct?
No. So this would be the RPK would be reads per kilobase. So that particular gene had a certain length, I don't know, 5,000 base pairs, and a certain number of reads. And if you divide that number of reads by the length, normalized to by the length in number of kilobase, you will get 250. So if the gene is 5 kilobases, then it means that that gene has in total 1,250 reads mapped to it. They don't have to be junction reads. The junction reads will be still a relatively small fraction. The reality of that example is that, which is also clear, yeah, this is actually an incredibly high coverage for a single cell experiment. So you will get a few genes that have this level of coverage. Most of them have this or this level of coverage,
So the point here is actually one of the main insecurity that you have is the fact that you are reading very short reads.
Yeah, that's one of the problems. Yeah.
So what about, and this is my second question, again, a very naive question. What about long reads technology?
Yeah, that will make all of these methods redundant. Yeah, probably. It's quite one of the frustrating things of working in genomics is that new technology comes and it takes a few years to come up with good analysis methods, and by then the technology has moved on in many cases. Now this is not the case yet because you do have a nanopore machine, but you still probably do the vast majority of your sequencing on short read machines. And across the world, the number of Illumina short read machines is so much higher than the number of long reads that I think there will be at least another 10 years when we will get the majority of short reads.
But this Oxford nanopore machine with long reads be something that in which you can, in some sense, validate these?
Yeah, potentially. Yeah, that would be. I mean, in a single cell, you can never validate. Yeah, because you kill the cell and then it's another cell. But yeah, I think you can validate these type of examples on single cell.
If I may.
Yep.
Yeah, on the Oxford machine and the nanopore, the problem is that it's really hard to do Oxford nanopore on single cell.
That's another problem.
When we do on bulk sequencing, we need 10 or even 100 more times the RNA that we use for the Illumina machine. So it's a really hard way to speculate for the single cell. Nevertheless, I believe that this is a really exciting way also for the bulk sequencing to have an idea. So one question, if I may, if you have a benchmark compared to other, I've seen MISO that is pretty old as method, but like if you have any idea of the comparison, like in RMATs for the bulk sequencing, not only for the
Yeah, so RMATs is for differential splicing, and we do compare with it in the context of BRI2. I've not shown it here because I had a limited number of reads. So BRI2 does differential splicing quantification because you basically treat the type of cell or the condition as one covariate, and you estimate the effect associated with that covariate. BRI1 instead does quantification of splicing, and we compared with Callisto. So Callisto on single cells in our hands did very poorly. And Callisto is a much more modern method, and you could try on salmon or these other derivations, but salmon is not conceptually different from Callisto. It's just a faster implementation, as far as I can tell. Or Sailfish, that's the other one that is very common. I don't know who I'm talking to, but
I'm not sorry. I don't know if you can see me.
Yeah, no, I can't see you because, or maybe I need to kind of do this. Yes, now I can. There you go.
Hi. Sorry if I'm not there. It's a really exciting and a really nice talk and really
thank you very much.
Just to build on what you have already seen, and I see that Stefano already take the point. The RPKM needs you to know the length of the gene to calculate, but for the
so we need the gene annotation. Yeah.
Yes, exactly. That was, and I suppose this is also something that in Callisto and Salmon that you cited is really affecting the results.
Absolutely, yeah.
So a better genome annotation that might be created through the long reads. This is one of the points. Might maybe help in the modeling. But I really like the way how you described the problem, because it's exactly what we need to do. Decomposition.
It's a greatly simplified picture because, of course, genes also have incredible variability in the transcription start site, for example, even more perhaps. And so we're really thinking in the simplified case where you have precise annotation and no variability in end or start or anything like that, and even the splicing are just exon skipping events when there's many more different types. It's just that things become much more complicated very quickly.
And just out of curiosity, because you talk about exon skipping, and I know working in sometimes in the splicing that the other phase that the people looking for the splicing is the intron retention. So have you tested for the intron retention there?
So intron retention, you could say, okay, the intron is now another type of exon, and we use exactly the same thing, and we do that with in the BRI2 paper. We use BRI2 also for RNA velocity. Now, I don't know if you know about RNA velocity, is this concept that tries to associate a dynamic feature vector to cells by looking at the ratio of unspliced to spliced reads. And then basically we say unspliced is another isoform, which is complete intron retention. We don't care which intron it comes from. But yeah, in principle, it's easily handleable.
Thanks, good.
So maybe it's a naive question, but I was wondering about the Bayesian vectors, not the
Yeah.
If you have a big amount of data, you have to clean most of the data.
Mm-hmm.
And then you have some observations like the peak observation, the big one, where there are very few, when there is very little information, there are just one or two.
Yeah.
But how do you make sure that your prior and your cleaning process are not influencing too much your results? I mean, there is very little information in the observation, and a lot of it's thrown away, and a lot is in the prior.
So the kind of pre-processing where you throw away things generally happens on a cell level. So you basically say, okay, I have quality control, and if a certain cell does not have a sufficient number of reads, I throw away all of that cell. Then I'm left with a pool of cells. Maybe I had done an experiment with 1,000 cells, and I'm left with 700, say, because 300 of them were just terrible. And then I'm left with 300 cells, and then 700 cells, and then typically what you would do is say, okay, I'm not going to consider all the genes because, I don't know, some genes I actually see reads only in 3 cells out of 700. So what can I say? Cut them out. That's the kind of pre-processing that you do. After alignment and all those very time-consuming efforts. What you're left is still, hopefully, a sufficiently large set of cells, each of them with a sufficiently large set of genes. So here I've kind of taken, because BRI is essentially a gene-centric, BRI1 is essentially gene-centric because the features it uses to impute features of the gene, I've focused on one gene. And across different cells, I basically said, okay, this is a good gene in that cell. But this very same gene in another cell is really not very good. Now, what I would do in BRI, though, I would use the good genes like this one to learn something that is, what are the signals in the sequence that are predictive of splicing in that cell. And so I'll use things learned on this cell to help the prediction on things that look like this, but are not this, because this is the same gene, but are in the same cell. So maybe I have 5,000 genes in each cell, and I have, I don't know, a few hundred that are good and enable me to do a reasonably good learning and predict on the remaining few thousands for that cell. And with BRI2 instead, we share both across cells and genes. Now, how can you evaluate whether the prior is good? There are methods in Bayesian theory. At some stage, I was quite excited by these methods, and then I think when you try to apply them to a complex systems as biology, you find out that all your priors are really bad. So in terms, all of these methods are basically, if I sample data from the prior, do I get something that looks like the real data? And actually, not really. You very rarely do, I think, because any prior is going to be so simplistic compared to the complexity of the cell system that it's not generating interesting simulated data from the prior. Nevertheless, I think the kind of signals that we aim to capture from genomics are still the really big signals, actually. So I don't think from genomics we can make super refined models that predict variations of 10%. No, we're really looking at things that are big changes. They're still hard to find because the amount of noise is very high and the data is very high dimensional. But and if you have big changes, then model mismatch, I mean, ultimately, yeah, it will matter a bit, but you still catch the big things. Alessio?
For example, if you have a good model, is it possible that this gives you information about features of the sequence itself that determines then the behavior during splicing?
What do you mean a good model?
A good model, for example, during performance.
But on which task? On predicting?
On predicting, yes.
Yeah, so the models that we were showing here, I mean, these were predictive models which were trained on bulk. So what you would do, you take bulk RNA-seq, you would use 70% of the genes as a training set and 30 as a prediction set, a test set, maybe some on validation. This was a neural network that tried to learn the percentage spliced in. And this was instead, I think, a support vector machine. The features were handcrafted in both cases. So that's one thing. So there were conservation scores, k-mers. K-mers are slightly handcrafted. They're not hugely handcrafted. But they weren't anything really complex. So there wasn't a real representation learning in it. I don't know. It's an interesting question. If you have what if you were trying to do an end-to-end and learn the features as well? It would be very hard to do it in a Bayesian way.